welcome. So this is our normal Thursday 4 p.m. seminar time, but today we have a very special seminar because we have many special guests with us. We have visitors from 3 different botanic gardens from across the country visiting as a part of our Rare Plant RaMP network. So thank you. Welcome to all of our RaMP participants. We've had a very filled week full of talks and workshops and activities. And I don't know, I've really enjoyed getting to know the RaMP mentees. So thank you all for all your great contributions. So we have 2 seminar speakers today, Allison Autry and Ava Joseph. And I'll be introducing our seminar speakers. And so first, I would like to introduce Allison. Allison is a PhD candidate in our graduate program. Alice comes to us with a wealth of information on orchids, having worked at a Longwood Botanic Garden in the past, working on ex situ conservation of orchids and very knowledgeable on the cultivation of orchids. And Allison came to us really wanting to get involved in conservation research and really to apply this to her passion, orchid diversity. So Allison has this really great project where she's looking at a rare orchid, the Spearanthes infernalis. And so she'll be telling us all about her research in conserving rare and disjunct orchids in a changing American West. Let's welcome Allison.
Hi guys, thank you everyone so much for being here. This is a great seminar turnout and I'm so happy to have our RaMP friends and colleagues with us here today. I'm going to talk to you, like Naomi said, a little bit about my research with orchids. Possibly not the first type of plant diversity that you think of when you consider California or the American West, but we certainly have plenty of them and the ones that we do have are really interesting. So about myself, like Naomi said, I'm a grad student here. I'm a lifelong orchid enthusiast. My first forays into the plant world actually came via growing tropical orchids as like a hobbyist. And I developed a love for them that eventually necessitated getting into that professionally. I'm consider myself a conservation focused researcher. And so all the work I'm going to be discussing with you all today is meant to work towards preserving the long-term viability and success of these species in the wild. And I'm currently doing my research in Eastern California and Nevada. So today I wanted to introduce some of you to the areas of interest in my project. Since I know we have guests from all over with us today, I'm just going to give a brief overview of the Mojave and Great Basin deserts, and then also talk about the alkali wetlands in the American West, where my orchid thrives that I'm studying, especially ash meadows, for those who might not yet be familiar. I'm going to mention some of the threats facing alkali wetlands, which are why conservation research like this is necessary and so important. I'm going to introduce you all to my study species, Spearanthes infernalis, discuss my research objectives, cover the preliminary results from my 2024 and 2025 field seasons, my future plans for Spearanthes infernalis research, and then a brief aside on Malaxis brachypoda, another orchid I'm studying. For the RaMP students that have been with me this week, I mentioned that in the past. So I'm going to give an overview today, and then after the seminar is over, I will bring in the plants themselves for show and tell as promised. So yes, but first Spearanthes infernalis, which sadly I don't have any insight at the garden with me yet, but maybe someday. I wanted to talk about the 2 main ecoregions that Spearanthes infernalis inhabits. So you can find it within the boundaries of the Mojave Desert. This is of course, notoriously dry area in the American West, primarily in Eastern California and Southern Nevada. It's home to some really iconic plants like the creosote bushes, the Joshua trees, the Mojave yuccas, and it gets very low annual average precipitation from 2 to 6 inches. And it includes famous habitats like Death Valley and Ash Meadows, which is the center of my research. Spearanthes infernalis also occurs within the boundaries of the Great Basin Desert. This is a slightly different climate. It gets a little bit more rain, but certainly not an abundant amount. It's more northern. It covers northeastern California, a large portion of Nevada, and then also kind of sneaks up into Idaho and Oregon a bit, as well as Utah. It is kind of dominated by sagebrush, so a lot of different Artemisia species, and pinyon juniper woodlands at higher elevations. So CalBG researchers spend a lot of time in both of these ecoregions. Also, for those who are not familiar with the landscape in the West Coast botany scene, I wanted to cover land management agencies really quickly, because we have a couple stakeholders at play when we're doing our research out in the West. The U.S. Fish and Wildlife Service and the Department of the Interior sometimes comes along with better mandates and protections when you're talking about national parks or wildlife refuge areas, because their mandate is more focused on conservation and preservation of ecosystems and resources. But we also spend a lot of time working with the U.S. Forest Service and the Bureau of Land Management, and these institutions have a much more mixed use mandate. So for them, conservation isn't always going to be the highest priority. Unfortunately, they lean a lot towards prioritizing logging, mining, and cattle ranching is a really big issue in a lot of the habitats we work in. So when I discuss the conservation threats facing my plants today, it's really going to be colored by which one of these land ownership boundaries they're under. And so I'll mention that as I discuss the different places I can find my rare species. I also wanted to talk about the alkali wetlands I'm in. This might be a habitat not all of you are familiar with. It's not incredibly common, and it's a result of two unique phenomena in the area that you find them, which is upwelling of groundwater and then massive evapotranspiration rates that really outstrip the precipitation that's received in the area. And what happens when you have that phenomena is you get these salt crusts on the surface of moist, or in some cases even mucky ground that has high water content, but also high content of minerals that come from this water that upwells from aquifers. And so it's a really interesting habitat for plants to try and survive in, because you do get a lot more moisture available to plants than in most parts of the deserts, but they also have some other challenges they have to face. And so we do a lot of research. These habitats are home to other interesting plants like the bird's beaks that we research here, the niterworts, the spring-loving centauries, that type of thing. And so these habitats are really, really crucial to understand and protect. And that's where I spend a lot of my summers. And it gets quite hot. I get really early starts to the day because summer high temperatures can easily exceed 100 degrees Fahrenheit in these environments. So Ash Meadows is a really interesting example of an alkali wetland in the American West. It gets much more evapotranspiration every year than it receives in rainfall, and the pools, wetlands, and marshy areas you see are all a result of groundwater from the really regionally important Amargosa River. It's the largest remaining oasis in the Mojave Desert, and there is a massive abundance of water here that supports really diverse habitats, including dune fields, alkali seeps, and groves of mesquite and ash trees. So it's this really beautiful, interesting place, but obviously any sort of groundwater extraction happening in the area can be a big threat to this habitat and all the species that live there. I did want to show you some of the residents of Ash Meadows other than my study species, because I think they're really cool. We have the Amargosa niterwort, Naomi's favorite plant, perhaps, depending on I guess which field site she's been to most recently. But we also have Zeltnera nematophylla, which is another really cool plant in the Gentianaceae family that's super charismatic, and actually all over basically any site where you can find Spiranthes infernalis, you'll be finding the spring-loving centaury as well. And then Ash Meadows is perhaps most famous outside of the botany community for its pupfish. The devil's hole pupfish is this really interesting plant that just grows in this one tiny little hole in the earth. And then there are also other species of pupfish you can find more widespread around the Ash Meadows area, like the Amargosa pupfish. And there are these really beautiful blue pools that are kind of centered in these marshes and are home to the pupfish. So that's just a little bit about Ash Meadows to set the scene for you guys. But unfortunately, these habitats are under threat. As I just alluded to, really anything that can affect groundwater levels is a pretty big threat to these habitats. So solar development and geothermal energy development in the area can cause huge problems. Lithium mining is a really big issue. Grazing and trampling by cattle can affect a lot of these riparian habitats, because as you can imagine, when you're free ranging your cattle in the Mojave or Great Basin deserts, they're gonna, be pretty desperate for water and sustenance, and they're gonna gravitate towards the riparian areas. Off-highway vehicle incursion can be a problem in areas that are less well protected. pathogen introduction can potentially destabilize these habitats. For anyone who's familiar with Mojave riparian areas, you might know about the screw bean mesquite, which has been really struggling lately, possibly due to pathogen introduction. Efforts to privatize public lands, of course, threaten any and all ecosystems that occur on our public lands. And so that's something that really colors our concerns as we're doing this work now. And climate change can be a big issue. You know, these areas are already incredibly hot and dry, and if they get hotter and drier, that can push even the really resilient plants that inhabit these places over the brink. So, skip the page. Now I want to introduce you guys to Spiranthes infерnalis, my study species. Initially, Spiranthes infernalis was believed to be endemic to ash meadows. That's where it was first discovered in the 1970s. It wasn't actually described as its own species until 1989. And up until recently, pending kind of the results of this research, it's still considered a Nevada endemic plant. Even now, we've only been able to find it at 2 different locations in Nevada, in Ash Meadows and then also in Railroad Valley, which is a whole 200 miles north and east of where it exists in Ash Meadows. And then it might have a potential 3rd location, which is Fish Slough, California, another alkali seep just north of Bishop, California, for those who are familiar with the Eastern Sierra area. This species is completely reliant on groundwater-fed alkali seeps and marshes for its survival, and not a lot has been studied in terms of its life history and ecology, but like many orchids, especially in Spiranthes, it is suspected to be selfing, possibly even an apomictic breeding system. It gets very high seed set rates, but not a lot of pollinator activity is observed. And it is dependent on relationships with different mycorrhizae to thrive. It needs mycorrhizae to germinate, and so recruitment can be a real challenge for these species. I wanted to show you all its phylogenetic placement right now. I know this is a lot to look at, but I'll try and kind of situate you all. On the right here, we have one of the more recent phylogenies of Spiranthes genus in North America as a whole. And as you can see, the Western North American clade kind of falls out basally here along this grade. And then once you get into a more detailed look at Western North American Spiranthes, as researched by Matthew Pace, who's the current expert on the phylogeny of this group, you see that Spiranthes infерnalis exists in this clade with another species, Spiranthes porifolia. They currently form an unresolved polytomy that I'm hoping I'll be able to resolve during the course of my research. But they're pretty clearly distinct in this phylogeny from all the other species, and that's pretty clearly reflected in the morphology of the plants as well. These 2 species both have really dense inflorescences relative to the rest of the group. So looking in a more detailed way at Spiranthes infерnalis and ash meadows is kind of this system of meadows and pools that pop up through the ground as the Amargosa River travels underneath. Like I said, this is the type locality of Spiranthes infernalis, and it occurs in a variety of different places in and around the Ash Meadows Wildlife Management Area. There are 4 main occurrences that I've been researching within the boundaries of the refuge, and then 2 significant occurrences I've discovered on BLM lands surrounding the refuge. Currently, the main threat to these plants is groundwater and aquifer depletion due to upstream mining projects. They're also located near the Western Solar Plan, and the plants that are on the BLM outside of the refuge are also subject to some cattle activity as well. But the main threat in this area is groundwater and aquifer depletion. And if you follow the news in this area, there have been a lot of proposed mining projects, more than close enough to this area to threaten all of the wildlife at Ash Meadows. In Railroad Valley, now we're up in the Great Basin Desert. We're not in the Mojave Desert anymore. And this is a more recent discovery, I guess a whole 5 years ago now, but pretty fresh. Thanks to Naomi spending some time out in Railroad Valley and just stumbling upon this plant, which had never been documented there before. Hundreds of miles disjunct from Ash Meadows, and there are 3 occurrences known from Railroad Valley now. It's a pretty large area, and these plants can be found on both sides of this big playa in the valley. The largest of these occurrences around a place called Chimney Hot Springs and Terrace Hot Springs, and there are hundreds of blooming stems at these populations. They're pretty consistently emerging year on year. They seem to be stable there, but they are threatened by cattle, and there's a lot of signs of trampling and herbivory anytime we go and visit these plants. Finally, I had mentioned that there are these mystery plants in Fish Slough, California. This is a really small population of plants that in the past had been identified as Spiranthes portfolia on herbarium vouchers, but they don't really line up with where you would ever expect Spiranthes portfolia to grow. That's usually a montane meadow species, but these are found in an alkali sea, kind of similar to where you would find Spiranthes infernalis. And so the identity of these plants have been called into question, and I have sampled them so I can do a genetic analysis and hopefully find out who they really are. So getting into my research objectives, the first of which, like I said, I'm going to be using molecular data, so DNA extraction and sequencing to better define both the population structure of Spiranthes infernalis of species, and then to also elucidate the taxonomic identity of the Spiranthes found in the Bishop area. I am going to collect and seed bank material from all 3 Spiranthes infernalis occurrences and develop protocols for ex situ propagation of these plants, which is an especial challenge given that they're orchids. I'm going to be surveying and have been surveying for additional Spiranthes infernalis populations in the areas of the main known occurrences. I've been collecting vouchers, and I've been making an effort to conduct demographic and life history studies in situ when I can. One example of this being that this year I collected root tissue material and sent off to Melissa McCormick with the Native Orchid Conservation Center out east to hopefully get sequenced so we can find out who the mycorrhizal affiliates of this species are. Finally, I want to use all of this data that I am going to generate during my dissertation research to write and implement a conservation plan and pursue legal protections for Spiranthes infernalis. So what this looks like is potentially listing the species under the Endangered Species Act. This is one of the strongest pieces of legislation we have left standing for protecting rare species and the habitats that they rely on to continue to exist. And I'm hoping that the data I produce with this project can allow us to generate a strong petition for listing the species. I also want to better understand the relatedness and the biodiversity of our main Spiranthes infernalis occurrences so that we can make better informed decisions when it comes to doing in situ and ex situ conservation actions for this species. And knowing the identity of the fish slough plants will really help with this, because if they are what we think they might be and they're Spiranthes infernalis, this will be a California state record. It could lead to potentially better protections for the fish slough area. The species would instantly be qualified for a really high rare plant ranking with the California Native Plant Society. And then we could potentially seek further protections for the area under pieces of legislation like the California Environmental Quality Act. So a brief overview of the population genetics work I'm going to be doing for those who might be uninitiated. We're basically using DNA extraction, sequencing, and then statistical analysis of the sequences that we receive to take a look at the genetic diversity and the relatedness of different occurrences and populations of the species. This is often utilized by researchers here at California Botanic Garden when we're looking at species with unusual distribution or species that are highly rare. And Spiranthes infernalis is both. It's only known from 2 occurrences and they're incredibly disjunct. So we have to figure out how that's impacted the gene flow and the diversity of the remaining individuals of the species. It can tell us how much genetic diversity the species has and how much is found within and between populations. And it can also give us inbreeding insights about inbreeding in the plants or the population structure. So a brief overview of what I accomplished in the 2024 season. That year, I sampled 196 total samples, the vast majority of which were either from the Railroad Valley Spiranthes infernalis occurrences or the Ash Meadows occurrences. And after that, I did in-house DNA extractions in the molecular lab here at California Botanic Garden. Here, I just show a couple stages in the process. We use a pretty standard CTAB protocol that works great on these orchids. And after that, we will use gels and cubiting to assess the efficacy of those extractions. Then we do library prep, and we have collaborators that we send those libraries out to be sequenced by. So we do that library preparation in-house, we send it out for sequencing, and then once we receive those sequences back, we use a variety of R packages to analyze our results. I was going to give you guys a brief overview of ddRadSeq, but Ava has a much better slide in her presentation about it, so I don't want to steal her thunder too much. But basically, we're just slicing DNA up with enzymes, attaching it to like DNA barcodes that will allow us to ID which sequences come from which samples. And then we sequence across many single nucleotide polymorphisms This type of analysis is really good for non-model organisms where we don't know which parts of the genome code for what. Without that reference genome available, a more randomized technique like ddRAD-seq is a good way to go. So I did a PCA next. This is a principal component analysis. Basically, a computer program transforms the data that I get from my sequencing onto a coordinate system, and the axes represent the principal components that differentiate the sequences from each other. So which ones basically contribute to the most variety in the data. This will allow us to visualize the genetic structure of a species or closely related species. It can also be used for, but it's a useful population genetics tool when it comes to differentiating how related different populations and subpopulations are. And here, as you can see, I've kind of got these color-coded circles happening for Railroad Valley plants versus Ash Meadows plants. The Railroad Valley plants were not differentiated from each other terribly on the first principal component axis, but on the second one that represented a bit less variation, they did clearly differentiate. And then as you can see, Ash Meadows and Railroad Valley were pretty cleanly differentiated. The subpopulations at Ash Meadows over here were not differentiated from each other much at all, which indicates that all those separate subpopulations I showed you guys in Ash Meadows are still experiencing gene flow between them, making it more of a singular large metapopulation instead of a place where you actually have distinct subpopulations. I was intrigued by the fact that this Railroad Valley population was split into two. I wasn't sure whether it was real or an artifact of coverage, but it also showed up on my structure plot, which I'm going to show you guys next. Or I guess let's hop to the structure plot because I want to talk about that first, and then we'll go back to those FST values. So the structure plot is another R program that we use to infer population structure. The K value is an important part of understanding the structure plot. It basically represents the number of clusters, and we have a program that tests to see which K value is kind of an ideal fit for the data. My data was best fit by K equals 2, which isn't deeply surprising when you consider that there are two main occurrences with a very large disjunct between them. But when you move up to K equals 3, which I've included here on this slide, you do get kind of this division in the Railroad Valley populations here, much more significantly than you get it in the Ash Meadows population. So that kind of lines up with what I was seeing on my PCA as well. But obviously the best fit and the strongest value is up here where we get this relatively clean split between the Ash Meadows occurrences and the Railroad Valley occurrences, which lines up with their massive disjunction. But the subpopulations did not distinguish very clearly on the structure plot either, just like they didn't, clearly differentiate from each other in the PCA. I'll head back to my FST now. This is called the pairwise fixation index, and it basically measures how differentiated two populations are from each other. So as the name implies, you're going to be looking at any two populations in your study on this one, trying to figure out how similar or different they are from each other. A high FST value indicates strong differentiation, and a low FST value indicates that they're more genetically similar. So looking at the results that I get on this, the populations from the same areas on pretty low FST values, relatively speaking. And then when you look at populations from different areas, they were incredibly high. This is essentially what you would have expected. And the one thing I wanted to point out is that I included the fish slough plants in this comparison, and they were the most related to an occurrence called Soda Springs at the north end of Ash Meadows. So that is the closest relative, at least in the study presently, to those plants. In the future, I'm hoping to look at other Sporanthes species on this metric or occurrences that I collected from other species to see which one the fish slough is most similar to once I kind of diversify what I'm looking at here. So moving on from that, I also took some population genetic statistics from these different occurrences. This is just a way to look at how much diversity and how much inbreeding is found in these populations, which can be a concern when you have smaller isolated populations of rare species like this. I calculated observed heterozygosity as well as expected heterozygosity, and then an inbreeding coefficient. I've kind of tried to color code here, different than the color coding that I've been using so far, and I do apologize for that. Red being observed heterozygosity that was in deficit of what was expected to be there, so things that are a little bit less diverse than you would hope to see, and then green coding for things that are slightly in excess of the amount of diversity you have expected to get. So that's a good thing. This is actually the type population here that's been split into two based on just like a roadway that was kind of going through the middle and dissecting the population. And the type population also happens to be the most heterozygous of all the populations I looked at. Thankfully, it is easy to access for seed banking purposes. And then for inbreeding, largely inbreeding was not a significant issue in these populations, but I did tag 2 in pink here that were a bit more inbred than the others. And then we actually had 1 negative inbreeding coefficient as well. So moving forward from my preliminary results that I generated in 2024, I wanted to talk to you guys about the field work that I've done so far this year. I've been surveying and sampling plants on Bureau of Land Management land around ash meadows. This is relevant as much for bureaucratic reasons as it is for biological reasons. Obviously, these plants are still really close to the populations that are within ash meadows, but they are in a different land management boundary. So thanks to the field work we've done this season, we now know that BLM has Spiranthes infernalis plants on their land as well as just the wildlife refuge. And so that's something we've been able to document this year. I've also been surveying for new occurrences in Railroad Valley, and I've been doing a lot of Spiranthes outgroup sampling in the area to better elucidate the identity of the fish slough plants as well. So those have been my 3 main objectives this field season. The results of my surveys for Spiranthes infernalis were as follows. I was able to find a new occurrence in Carson Slough on Bureau of Land Management land just outside of Ash Meadows. I collected 50 new tissue samples from the Ash Meadows area across 3 occurrences this year, 2 of which were on Bureau of Land Management land. I also undertook some surveys guided by Sentinel-2 satellite data in Railroad Valley, finding 2 new occurrences, and I collected 40 additional tissue samples across both of these. And overall, it was a pretty good season in terms of what we found out about the abundance of Spiranthes infernalis, right? We found over 100 new plants documented over multiple localities. So this is good news for the long-term survival of the species and also provided me with more plant material to undertake my population genetics research on the species. So hopefully it might make a little bit clearer, why some of these splits in Railroad Valley are happening and that type of thing. These are the new occurrences in Railroad Valley. I wanted to give you guys kind of a sense of scale. Each of these are separated by a couple miles. Railroad Valley is quite a massive place. Chimney and Terrace Hot Springs is still the largest known population in this area, and is the population that's been known the longest since 2020. But this year in 2025, we found another population of at least 100 plants, I would say. It can be kind of hard to get an exact count when you're working in really graminoid-rich areas around Locks and Reynolds Spring. And then a small and highly cattle-compromised population in a place called Bullwhacker Springs on the other side of the valley. How do you find these plants? I wanted to give you guys kind of an insight into what the surveying process looks like when you're out in the field looking for Spiranthes infernalis, and it can be challenging. You're working in really beautiful, lush, but also reed dense plant vegetation habitats like these, where you have a ton of anamopsis, rushes, grasses, and the like. And Spiranthes infernalis plants are largely not that tall. They rarely will exceed a foot on a really, strong, robust plant. And unless they're in peak bloom, they kind of look like tadpoles. That's what I describe this phase as, the tadpole phase. So it can be a little bit of a challenge trying to pick these plants out of a literal field of grass. Definitely gives new meaning to the phrase needle in a haystack. And so we've been using something called Sentinel-2 satellite imagery to guide our surveys. The grasses are a little bit less sensitive to moisture than the Spiranthes are. So what we can often do is identify really moisture-rich areas on this imaging that we can then target for Spiranthes surveys. So for example, these are the plants at Chimney and Terrace Hot Springs right here. You can see the plant occurrences overlaid on the satellite imagery and how they really stick to that super dark area on the map. So a lighter blue area like this might not be suitable, but when you see these little kind of potholes of high moisture on the map, that might be somewhere you would want to target. So once we finished our time out in Railroad Valley, I moved on to thinking about the plants and fish slough this summer. So I am basically reconstructing the Western US Spiranthes phylogeny to find a place for the fish slough plants in this phylogeny. I've been sampling a variety of species. The main four that I'm looking at being Spiranthes infernalis, of course, my study species. Spiranthes porifolia, which is its closest relative and the one that it currently sits in an unresolved polytomy with on the Matthew Pace Spiranthes phylogeny. And then also sometimes on purpose and sometimes through misidentification in the official CCH2 records, I've been sampling Spiranthes extellata and Spiranthes romans-affiana as well. So these are two of the closest relatives to this branch of the Spiranthes phylogeny here, where porifolia and infernalis sit, and will kind of serve as my outgroups while I'm comparing individuals from these two species to each other genetically. I collected 57 tissue samples this summer in Spiranthes taxa from, it looks like we covered up our number, but there were, I think, about 10 populations and over 3 taxa that I looked at. And I confirmed 7 populations of true Spiranthes porifolia in California this year and sampled 5 of them. This is a big deal because Spiranthes porifolia recently was redefined in the Jepson. Up until the late 2010s. Anything in California that wasn't identified as Spiranthes romans-affiana was Spiranthes porifolia, and that recently changed. So if you want to figure out whether something that was identified as Spiranthes porifolia was actually that species, you kind of have to go in and look at the records themselves, the vouchers, any pictures that were uploaded from the area, or just go there in person and see how it keys out on the new key. But we were actually able to find some true Spiranthes porifolia to compare to our Spiranthes infernalis this field season. And so I'm hoping that is going to lead to a couple new products this season, one including a phylogenetic tree that has Spiranthes infernalis, Spiranthes porifolia, and then romans-affiana and extellatus outgroups on it, which should hopefully lead to a resolution of the Spiranthes porifolia and Spiranthes infernalis polytomy that's happening there. And will help me place finally my fish slough mystery plants on a phylogenetic tree and determine what they are. I also am hoping to produce a much better population genetic analysis of Spiranthes infernalis itself this year, utilizing the increased sampling from ash meadows that I undertook this year, as well as the new Railroad Valley occurrences. And to do this, I have 150 samples of DNA that I will be extracting this fall and winter. That concludes my Spiranthes infernalis update. I also wanted to take a moment to talk about Malaxis brachypoda. To the RaMP students that have been with me this week, you've kind of already been introduced to this species, but this is what it was originally supposed to be my study species for my orchid research here. It is this really interesting disjunct population of a largely otherwise circumboreal species that is known from the Great Lakes region in upstate New York in North America, as well as the far northern Canadian Rockies, but it is found in Colorado and California at really high elevations. My theory on this is that it was kind of a climate relic species from pre-Holocene warming times, and as the most of its range retreated northwards, some plants were kind of left in these high elevation refugia. And I was really hoping to study the population genetics of this species in California, but unfortunately, it's kind of on its way towards extirpation in the state. And so I'm more documenting that now that I am conducting a full-scale population study. There was only one population that had been documented in California recently, and it is in now massive decline following recent wildfires. I've gone to all the other places it's been documented, including a long extirpated population in the San Jacintos, just to look for it. And I was able to find a total of 0 blooming plants in situ this year. In total, since I started looking for this plant, I found 2. And so it's really doing badly. It's currently California rare plant rank of 2B.1, but I'm afraid it is heading towards 2A.1 at a pretty rapid pace. And so, like I said, I can't do population genetic study on these plants because there are no populations remaining in the state, but I have not abandoned it from a conservation perspective. There are ongoing propagation efforts, and I have flasks of this plant that I propagated from seed in my office that, as promised, I will be bringing by for you all to take a look at once the seminar is over. That is my last update for you all today. I think we'll hold questions for the end, maybe just so Ava has time, or would you rather?
We get to a couple quick questions.
A couple questions now if anyone has any, and then I'll hand it off to Ava.
So now it's my pleasure to introduce Ava Joseph. Ava is an alum of the RaMP program. So Ava just completed her RaMP year last year, and she's fresh. So she's fresh out of the RaMP program and newly joining our graduate program as a PhD student. So this is Ava's first semester in graduate school. And so Ava's presentation will be the product of her RaMP research. So that's really exciting to see the culmination of year-long research, a lot of hard work. So Ava's talk is on if inflorescence morphology is not indicative of relationships in Eriogonum kennedyi complex.
Cool. Awesome. Yeah, so I'm really excited to talk to you all about my research over the past year, especially for this RaMP group. And I studied the Eriogonum kennedyi complex. And yeah, inflorescence morphology is not indicative of relationships. So what does that mean? We'll get into it. Thank you. Just to situate everybody, we are in the family Polygonaceae in the genus Eriogonum, which is the wild buckwheat genus. These plants have a mainly Western North American distribution and are incredibly speciose, having over 250 species described, and are also incredibly diverse, both morphologically and in the habitats and niches that they occupy. These plants all have 6 sepals and usually have 9 stamens, and their flowers are held in involucres, which are these little cup-like structures shown here. All right, and so prior phylogenetic studies that included what I'm calling the Eriogonum kennedyi complex start with this Elizabeth Kempton's 2012 paper on the systematics of the subgenus Eriogonoidea. She used 3 loci in this analysis, and she revealed this well-supported clade containing both Eriogonum kennedyi and Eriogonum wrightii, although the relationships between the taxa were not well resolved, and only one of the 5 kennedyi varieties was included. So moving forward a couple years, Peter Perriman and others in 2021 did a next-gen sequencing of the tribe Eriogonae. Like Kempton, this showed a well-supported sister relationship between kennedyi and wrightii, but once again, only one of the 5 kennedyi varieties was included. So talking about Eriogonum kennedyi, what is that plant? So these are perennials. They form these low cushion-like mats. Their leaves are really densely clustered together. They can vary in shade and size, going from like small and linear to maybe like slightly larger and more rounded. Their inflorescences are composed of this long stalk-like scape, and then all of their flowers are clustered together at the very top into this capitate head. And I have a map there on the right of occurrence records taken from the Consortium of California Herbaria for each of the taxa in this complex that I'll be going through when I describe each one by one. Okay, so the first 2 varieties I want to talk about are Kennedyi ashmontanum and Kennedyi kennedyi. So ashmontanum has this federally threatened status, so it has that federal protection. Kennedyi does not. It's more like common, widespread. These 2 varieties both have different type localities within the transverse ranges, and yet they are thought to both co-occur in the same areas. However, this is really hard to distinguish because the main differentiator between these 2 varieties is in the scape surface. So kennedyi is thought to have a glabrous scape, whereas ashmontanum is thought to have a tomentose scape, meaning it's like hairy. But this is not always accurate and can be really tricky to use, especially because the hairs can fall off and all that. So finding out where ashmontanum actually is found is really important because of that federal protection. The next 2 varieties I want to talk about, Purpussii and Panicula, both occur in the southern Sierra Nevadas of California. Purpussii is widespread throughout this area and just barely crosses the border into Nevada. Panicula is considered rare in California and is only known from a small handful of occurrences down at that very southern portion of the range. All right, and then our last variety of kennedyi is Alpiginum. So this is also considered rare in California. It only occurs in the Transverse Range Alpine, which are, very few alpine regions in the Transverse Ranges, and these are over 2,500 meters. And because it's a high elevation plant, it has very reduced scapes compared to the other kennedyi varieties. And then the last taxon in this complex is Eriogonum variety subscaposum. This plant is widespread throughout California and down into Baja California. It's a super common montane species and can be found at mid elevations all the way into the alpine. And basically anywhere you find kennedyi, like a couple meters away, you're likely to find Eriogonum variety, which can be tricky because they look very similar. They are also perennials that form these low cushion-like mats, and their leaves can look very similar at times. But the big differentiator between these two is in their inflorescence shape. Radii subscaposum has this branching form. All right, so now just to distinguish the two even more, Eriogonum kennedyi has those 5 varieties I just talked about. It has the capitate inflorescence form. It only grows on these exposed and rocky habitats, such as the photoplankton environments in the San Bernardino Mountains, and it flowers in early summer. Whereas Eriogonum radii subscaposum has the branched inflorescence form. It's a habitat generalist, growing, even under the understory of trees, and is widespread throughout the state, and it flowers in late summer. Yet despite these differences, they can grow within inches of one another. So that is really, hard at times, because if you don't have either flowering stalks or old flowering stalks, it's really tricky to tell the two little cushions apart from one another. And it's also raises concerns because of this phenomenon, where this random branching is occasionally observed in the Eriogonum kennedyi varieties. I myself and other botanists have observed it in almost all of them except for the Alpichinum. And some botanists have described them as kennedyi radii subscaposum hybrids. So in this project, we sequenced some of these Ashtamontanum intermediates to see whether they fall out as hybrids or if they're a new taxon or what's going on with that. Okay, so to get into how I actually went about doing this project, I went into the field to collect tissue samples for DNA extraction, as well as pressing plants for herbarium vouchers, and then also preserving some flowers and leaf tissue for anatomical work. Also just taking note of how the plants are growing in their environment as well. And then back in the lab, I did the DNA extraction and then preparing the libraries for sequencing, and also did the analysis. So, okay, the sequencing method we used, ddRADseq, like Allison gave you guys a primer on. So this is a next-gen sequencing method that sequences thousands of loci from across the genome. It involves a double restriction digestion. So it cuts out 2 points of the DNA, and then with those fragments, you ligate adapters and barcodes to them. You then pull them all into one tube, do size selection, PCR, more size selection, and then sequencing. They get sequenced through Illumina NextSeq. And then for the analysis, I used iPyrad 2D multiplex and assemble all of these fragments, and then IQ-tree to get the maximum likelihood estimate. Okay, so now we get to see the results. So this is my maximum likelihood tree. The red circles indicate bootstrap support values that are lower than 95%. The triangles indicate clades that I've collapsed just for simplicity's sake. And then I've color-coded the kennedyi samples in green and the radii subscaposum in that yellow. And right off the bat, you can see that they are not clustering together, meaning they're not monophyletic, which is super interesting. But we're going to go through this tree bottom to top, step by step with you all. So starting at the bottom, our outgroup is Eriogonum elongatum. And then I also included 2 other varieties of radii, those being Membranaceum and Dentatum, just to situate this complex within the larger radii group. And then above those outgroups, we have our first clade of radii subscaposum. Above that, we have a clade of Kennedyi ashtamontanum and kennedyi from that kennedyi type locality. The ashtamontanum samples are indicated by the arrows, and these are not clustering together, rather they are just coming out intermixed with the other kennedyi. So because of that, and because we're in that type locality, it's likely that these sampled individuals are just representing samples of Eriogonum kennedyi variety kennedyi. So above that, our next clade includes Kennedyi purpursii and Kennedyi panicola. Again, those arrows pointing to the tips are showing where the variety panicola samples are falling out. And these are also not coming out as a monophyletic group. But more samples of panicola have been collected that I wasn't able to include in this project. So more work still has yet to be done to determine if this is like actually a valid taxon or not. And then above that clade, we have our second clade of Radii subscaposum. Then moving into the top half of the tree, we have yet another clade of Radii subscaposum, a second clade of Kennedyi variety kennedyi. And then above that, we have our alpine variety Kennedyi alpiginum. And then finally, at the very top, we have our potential hybrid intermediates, and then Kennedyi variety ashtamontanum from its type locality. So again, the arrows with the tips are showing where those intermediates are falling out, and they're not clustering together. They're not in any position that would indicate that they're any sort of hybrid. Rather, they're just falling out intermixed with the other Kennedyi ashtamontanum. So because of that, we can confidently say that there's like these like intermediate or individuals are not actually like a hybrid or new taxon. So here's that whole tree again with, you know, what I just went over, all of the different taxa. And you may be wondering at this point, why are there like these three different clades of Radii subscaposum? Why are they not falling out together? What's going on? And that's because there is some super strong geographic signal with this tree. So in that inset map, I've indicated those 4 different like mountain range regions with those 4 shapes. And these map on super cleanly to where the samples were collected in each clade. So the Radii subscaposum down here, these samples were all collected from the San Gabriel Mountains. These Radii subscaposum were all collected from the Sierra Nevadas. These ones are all from the San Bernardinos. So that's super interesting. The only outlier to this pattern are the Kennedyi alpiginums, which are all grouping together despite being from the 3 different like mountain peaks. Getting back to my title on inflorescence morphology, I was really interested to look at this transition from the branched inflorescence form to the capitate form. So Eriogonum elongatum, and which was our outgroup, and then all of the other Eriogonum radii have the branched inflorescence form, so that's assumed to be the ancestral state. And then all of the kennedyi have the capitate form, so that is assumed to be the derived state. And looking at this, there are potentially 3 different instances of that capitate form being derived. So that's really interesting to see and to think about, potentially in these 3 different mountain regions, a unique occurrence of the capitate form arising. So yeah. And then I just wanted to throw in some preliminary structure work. So Allison kind of explained structure, what that analysis is earlier in her talk, but all of the different clades on the tree are represented at the bottom, and then the shapes kind of like are organizing them into their geographic like bins. And just looking at our ashtamontanum, which is our federally threatened variety, compared to the other 2 clades of kennedyi, you can see that here's the federally threatened one compared to the other one from the San Bernardino Mountains. It's a little bit more distinct, but then looking at this one from the like Ventura County area is a lot more like genetically distinct from even this like these other 2. So that's really interesting to see this like genetic signal there. And then looking at the Radii subscaposums in those 3 different clades, these are also surprisingly distinct. So, this one from the San Bernardino Mountains and, this far one from the San, Gabriel Mountains, are fairly similar genetically, which makes sense because of their close geographic proximity. But then if you look at these Sierra Nevada ones, they are like incredibly different, which is, just really interesting result to get. So, yeah. So my conclusions, for this project are that, most of the Eriogonum kennedyi varieties are monophyletic, although the species as a whole is not monophyletic. Eriogonum rhytii subsposum is likewise not monophyletic. And then we found that federally threatened, Kennedyi ashramontanum is limited to one locality, that being the San Bernardino Mountains. My next steps are to continue testing for introgression, improving my analyses and getting them like publication ready. And then I am now in the role of being a PhD student. So that has been a lot of fun and a lot of work as well. So, yeah. And then I just want to acknowledge everyone that helped with collecting, with doing preliminary lab work and analyses, Holly and Clay at UCR for sequencing, everyone in my RaMP cohort, and then the NSF and US Fish and Wildlife for providing funding for this project. And then because we're at the RaMP Symposium, I just want to throw this out there and say, thank you, RaMP. I am like so incredibly grateful to have had this opportunity to like be a RaMP scholar and just like do all of these like incredible experiences, like presenting posters, giving talks, doing workshops, and then having this cohort of like young scientists that you can like be in like a little group together. So just congratulations to all the new RaMP scholars. I'm so excited to see what you guys do next. So yeah, thank you.
That was great, Eva. Thank you. We have time for a couple of questions.
How do you fix this in terms of names?
I'm afraid I'm gonna do that like horrible thing where I put out the molecular work and then don't do any of the taxonomic work that needs to be done. Just because I don't know what to do. So maybe like some more work in the future. Someone can like unravel what's going on with these, yeah.
So far, it seems like you've been discovering more so than along species lines. It breaks up during this like geographic molecular signal. Do you know at what point that stops? Like, are there more species that you expect would be mixed in like this, or is it really just these two SoCal cushion buckwheats that are going to be this way?
Like other buckwheats in the area that might also be
Exactly. Like, are there
this
other gnat buckwheats with like kind of overlapping distributions that you think would also fall victim to this?
I think so, because these are kind of like the like Southern California mats. As you get into like Northern California, you get other like Cushing buckwheats, and those kind of like fall out like separately. So I think it's kind of just like a common morphology that arises.
So at the very least, this should only be affecting kennedyi and rhytii, hopefully.
Yeah,
Okay.
I think so. Yeah.
Okay.
Yeah.
It's good to know.
And then you've got the fact that they're variously endangered.
Right. Yeah.
Yeah.
Yeah. So that's kind of why
You should fix the nomenclature.
I'm a little hesitant to fix the nomenclature because of, I'm confident of Kennedyi ashramontanum being its own unique thing and maintaining that like federal protection, but it's like hard to think about like where everything else falls out after that. So yeah. Awesome. Cool.